So up until this point, we've talked a lot about DNA, and it's one of the nucleic acids, and now we're going to talk a little bit about RNA. Um, like DNA, RNA is a string of nucleotides. Right? And we can really view RNA as the middleman in the process of creating a protein from a gene in DNA. So RNA, right, helps create a protein from DNA. Now, I do like this table quite a bit because it shows you the differences in RNA and DNA. So you'll see here RNA is much smaller, DNA is much larger. RNA is a single strand nucleic acid, DNA is a double strand. Now, there are three types of RNA. And these are the types, they're the main types found in the cell, really involved in transforming DNA into a protein sequence. So, again, we're transforming DNA into a protein sequence, and that's what we're focused on. And so, the first type is messenger RNA, which we're going to abbreviate as mRNA. The second type is ribosomal RNA, which we are going to abbreviate as rRNA. And then we are going to have transfer RNA, which we are going to abbreviate as tRNA. So we're going to talk about each of these and how RNA helps with protein synthesis. So when we come over here and look, we're going to start with a process called transcription. And basically, the process of making a protein from DNA has two key steps. The first step is to make a gene copy from the DNA found in the cell nucleus. And so in transcription, what happens is the DNA's double helix unwinds so that a complementary copy can be made from one strand. Okay, and so we have basically what's going on here. Another way to say this is that mRNA is transcribed from one strand of genetic DNA, where there's one strand of DNA is used as a template, and then we make a copy using the RNA. Okay, and so when we look at this, the copy that's made is the messenger RNA. Okay. Now, this is a single-stranded piece of RNA, and it contains the bases complementary to the original DNA strand. Now, gene copying is catalyzed by RNA polymerase. And so I want us to visualize and look at the process here. So what we see here is this large enzyme, it's RNA polymerase, right? And here we have the DNA. So the DNA comes into the RNA polymerase and it gets unwound, right? And so we have a strand, a template strand here, and then this is just the non-template strand, and it's just going to hang out. And so then what happens here at the template strand is that you've got these free RNA nucleotides, and they come over and they match up. So everywhere there's a thymine, right, we match with a adenine, and then we see the cytosine and the guanine, and we match everything up. Now, notice when there's an adenine here, right, we match up with a uracil here. So you're not going to end up with any thymines in the RNA strand. Now, what happens then is that newly made RNA strand leaves, and then the DNA comes back together and it also leaves. And so what happens here then is this RNA travels, helpful if I could spell travels, from the nucleus, because all this is going on in the nucleus, to the ribosome. Okay, so now we're heading to the ribosome. And so I want to remind you that the ribosome is an organelle, right, that's composed of ribosomal RNA or RNA and protein. So we're basically going to think of this as a protein factory. Okay, and it's just the place where the nucleotide sequence of the mRNA is interpreted into an amino acid sequence. Now I'm going to abbreviate amino acid as AA, like I've done before. Okay. Now, the ribosome has two RNA protein subunits, and they're called the small subunit and the large subunit. Okay. And the mRNA strand fits into a groove on the small subunit with the bases pointing towards the large subunit here. Now, the second step in protein synthesis, which is still kind of going on here in the ribosome, right, is translation. Right? Now, the facilitator of this process is transfer RNA. Okay. There's several areas on the tRNA sequence where the complementary bases can hydrogen bond. And so a few things that we see here when we talk about transfer RNA is this is a kind of a blown up cartoon of it, and you can see there's hydrogen bonding here between these kind of base pairs. And so what happens is this hydrogen bonding gives the tRNA a really tightly compacted T-shaped structure. Now the tRNA has a 3-base sequence. It's an anticodon at its anticodon loop. And so when in the ribosome, the anticodon can hydrogen bond to the complementary, sorry, I pointed to the wrong thing. Here's the anticodon. So the anticodon can bond to the complementary pairs here in the mRNA. The tRNA also has an acceptor stem where it binds an amino acid, and this is the acceptor stem. Sorry, I got ahead of myself there. So acceptor stem here, and then the anticodon here. Now, the only way to get an amino acid incorporated into a growing protein chain is by bringing it to the ribosome bonded to the tRNA. Okay, so the tRNA is gonna bring the amino acids. There's your amino acid here. Okay, each of the 20 amino acids has one or more tRNAs just available, hanging out to bring the amino acids to the ribosome here. So let's talk about these codes and how each amino acid is signaled to come over to be part of the protein. So here is the different mRNA triplets are called codons. So let's start there. So a given mRNA triplet is called a codon. Okay. And basically what that codon does is you have the mRNA, it's transcribed from the DNA, and it contains a sequence specific to the protein that needs to be made using these codons. Okay, so for example, right, let's say we want to know what is the sequence of amino acids from the codons in mRNA below, right? And so let's say we have, this is the 5', we have CAA, that's our first codon, and then we're going to use a parallel kind of slash here to separate from AUC and another slash, and then GUA, and then we have the 3' in there. And so what we do now is that we come over here and we say, all right, which amino acid comes from this combination? So we start in the middle here, and that's kind of where this 5' is. So we say, okay, here's the C, and then the next thing I do is move to the A, and then I move to this A here, and then we have glutamine, GLN. And so we come here and we say, okay, we've got GLN. Then between the amino acids, we use dashes, and then we say, okay, now we're going to start with A, and then we are going to go to U, and then we're going to come down here to C, and that gives us isoleucine. And so now I'm going to write isoleucine here. Right, and then we're going to do another dash, and then we're going to come here and we're going to look at G, and then we're going to go to U, and then we're going to come here to A, and we're going to write valine. And so that's how, oops, sorry, I wrote all the valine there. That's how we can go from the mRNA sequence to the protein sequence. You have 64 codon combinations are possible from the 4 bases, and the codons that you're going to notice that are UGA, UAA, and UAG are stop signals. And you are going to see those stop signals here, here, and here, but we see everything else kind of codes for different amino acids there. And we start with a 5' and move to the 3'. Now, there's another question that you could be asked, so I want to mention that to you. You might be asked to provide the mRNA and the protein sequence from a DNA strand. And if you're asked for that, what you're going to do is you're going to come here to the 3' end. You're going to use TAA. So here's the DNA strand. We're still part of the okay. So there's the DNA strand. Now, each triplet in DNA has a complementary RNA strand. So for example, if we have T, right, it's going to be A, and then the A is the U and the U, right? Now we're starting with a 5', and then we come here, we have G, G, C, and then we have C, G, C, and then we have U, U, A, and then 3'. So that is the complementary RNA strand. And then from this RNA strand, now we can come in and get the amino acids, okay? And so if there was a little skip or pause there, I got interrupted. So just if it sounded funny, that's what was going on there, but you haven't missed anything. Okay, so we've got all of our RNA strands here. And so this is what we're going to use to get the proteins. So we start here with A, and then we go to U, and then we go to U, and we see that it's isoleucine. So I, L, E, right? And then we're going to do a dash. And so then the next one we see is we come to G, G, and then C, and then we see glycine. Right? Then we are going to come to C, G, C, and we see arginine. And then we are going to come to U, U, A, and that gives us leucine. Okay. So these are two different kind of questions that you can be asked as far as using this wheel to either figure out the amino acid sequence from mRNA or the amino acid sequence from a DNA template strand. So I want to review this process a little bit and look at the picture, okay? So here is a really beautiful image of kind of the big picture here. So when we look at this, right, we are over here in the nucleus, and it's the site of transcription. And what we see is that DNA, right, is going to unwind in the RNA polymerase, okay? And the complementary copy of the DNA template, right, which is right here, right, is called the mRNA, and that's created again in the RNA polymerase. Now, that mRNA, right, is going to leave the nucleus, and it's going to attach to a ribosome, and translation's going to begin, okay? So we are leaving the nucleus. Now, before the tRNA can be used in the ribosome, an amino acid also over here. So mRNA is left, and while mRNA is gone, we've got tRNA over here, and tRNA is picking up amino acids. Okay, so tRNA is picking up amino acids, and at the same time, it's kind of traveling over here to the ribosome. Now, that means then that these amino acids are ready to be used in protein synthesis. So the protein synthesis begins when mRNA kind of positions itself here at the ribosome, right? And you have a first codon in mRNA is the start codon, which is AUG. So AUG is the start codon. Okay, so we're ready to start. And then an activated tRNA with the anticodon of UAC comes and is attached to methionine, and that bonds to the mRNA, right? And then we undergo the process of protein synthesis. And so as the ribosome moves along, the mRNA, a new amino acid forms a peptide bond, and we grow the polypeptide chain here. We see the activated tRNA hydrogen bonds, right, to its complementary. And so it just kind of, these come in, they pop on, right? And then we're building the peptide chain here. And then the tRNA re-enters the cytoplasmic pool of free tRNA ready to be used again. So it just kind of comes along. Now, eventually, the ribosome is going to encounter a stop codon, and the protein synthesis is going to end. When that happens, the polypeptide chain's released from the ribosome. Okay, so this will be released, and then The initial amino acid methionine is also often removed from the beginning of the polypeptide. So that kind of signaling start amino acid gets removed. And then the growing polypeptide will fold into its tertiary structure with any of its disulfide links or salt bridges, and it will make a biologically active protein that will go on and do many things. So this is basically, in a snapshot or a review, how proteins are made. Now, I'm sure the burning question in your heart is, what happens if there is a screw up, right? Well, that's called a mutation, and that's just a change in the DNA sequence. And sometimes we can have silent mutations, right? And when we talk about silent mutations, these are mutations that occur but don't affect the function of the protein. So when we talk about this, one of the things that we may see is no change in the protein sequence at all. Right? It's important to understand that about 2.5% of the DNA in your chromosomes encodes for proteins, and the rest of the DNA is non-gene, or commonly referred to as kind of junk DNA. Even though it's not junk, it does do things, it just doesn't encode for proteins. Now, there's more than one codon that codes for each amino acid, right? So, for example, if we had UUU, okay, and it was accidentally changed to UUC. When we look at this, right, UUU codes for phenolphthalein, right? But UUC would also code for phenolphthalein. So if there was that change, the polypeptide and the peptide and the protein would still function the same way because it's the same amino acid. Now, there's kind of a second type we can see here where a change in the protein sequence occurs. But it has no effect on protein function. And so, for example, if the codon AUU mutated to GUU, right, then the amino acid isoleucine, right, so if we come here and we go, all right, we got AUU, so that gives us isoleucine, ILE, was mutated to GUU, so we're over here GUU, and now we're valine. There's not going to be much of a change. The two amino acids here have are similar in polarity and size, and so they just don't have much effect on the protein. So there's not much effect on the function of a protein. Okay. And so those are kind of two examples of silent mutations. Now, you can have mutations that are not silent. And these would be a change in the protein sequence that affects the protein function. So, for example, if AUU were mutated to AAU, basically that what would happen is that isoleucine would change now to sparginine. So when we look here, we would say, okay, hey, look, if we have AAU right here at asparginine, which is ASN, and that's a really big change. This is a nonpolar amino acid, and it's replaced with a polar amino acid. And now, right, that's going to be a really big change, and it could change the structure of the protein. And if the structure of the protein is changed, then the function of the protein can also be changed. So overall, rarely when DNA replicates do errors occur. Every so often they will occur, and that's just a spontaneous mutation. Environmental agents that produce mutations in DNA are mutagens, right? And many mutagens are carcinogens, which are cancer causing. Right? Viruses can also cause mutations. Here are some common protein or chromosomal defects, and then the disease symptoms. And so there are kind of these genetically inherited diseases that you can see here, and they are produced by a lot of times some sort of mutation, or they are inherited. If you are interested in this, you'll cover this a little bit more if you are a science major in genetics, right? If you are not a science major but a nursing major, you may see it more in microbiology as well. So that ends our screencast on protein synthesis, and then what happens when there are errors in protein synthesis. And then we are going to finish up with our third screencast on viruses. And so that's it.